everybody so much for joining us. It is Translational Science Week at the CTSC of Northern Ohio, and we appreciate you joining us for another event that focuses on safety in research. We are delighted today to discuss safety in research and safety with IRB in our clinical trials. And we have a special guest today. She is a former Annual Pilot Awardee through the CTSC and a pediatrician at Rainbow Babies Hospitals. So Dr. Lydia Furman is here to join us today. She'll be talking to us about her experiences sitting on the IRB review board and, of course, clinical trials in general. At the CTSC Northern Ohio, we are a collective of 7 institutions. So we all work together through collaboration. That is our main goal in helping train and educate the next generation of researchers. We do that through kind of 5 key components, being resources and services, workforce development, community engagement, health informatics, and our clinical and translational funding opportunities. We've got a variety of open opportunities with our funding right now, our annual pilot, and tomorrow, as a part of Translational Science Week, we are releasing another RFA for our themed pilot. So if you are a member of the CTSC, you'll have an option to receive that RFA tomorrow morning. If you're not a member, you'll have an opportunity to join at the end of this webinar, of course, as we close out all of our webinars with that. But again, exciting opportunities to come in our training. This is part of that, offering these opportunities to connect with our investigators on issues that they are engaged with and intrigued with the most. So our recruitment opportunities really allow us to focus on hot topics, so to speak. And so we have heard that this is a really intriguing topic, not only to investigators putting on clinical trials, but all of the people who work in that realm. So thank you very much, Dr. Furman, for joining us today. I'm going to pass it over to you and look forward to hearing your presentation.
Okay. Thank you very much, Laura. I appreciate that intro. I'm going to slip into slide mode here. Okay. Well, thank you for coming today, and I'm very glad that I'm able to be here. I just ended jury duty this week, so I'm going to talk about safety in research. Happy birthday, CTSC. I think it's a wonderful week of celebrations, and hats off to those who organize it, and that includes you, Laura. Wow, lot going on. I have no relevant disclosures or conflicts of interest. I did use Google's AI to inform parts of this presentation. My goal is, I think I'll have easily 10 minutes at the end for questions. So if it's okay, just jot them down on a piece of paper, tuck them into the chat, and I'll make sure and answer them, I hope. Our objectives today, I'd like you to be able to list the sections of the Code of Federal Regulations that are most relevant to participant safety protection. I'd like you to be able to state how the Code of Federal Regulations is operationalized to help study teams protect the safety of research participants. And I'd like you to be able to state potential risks to research study team members and how those could be mitigated. But let's talk about participant safety first, and that's always job one, whether you're a study team or an institutional review board. I'd like to frame the discussion today by beginning with the Belmont Report. You likely know that this is the ethical underpinning for the human subjects research that we do. It has 3 pillars. The first is respect for persons, which respects the autonomy of each individual, their ability to make a decision for themselves, and protects the autonomy of those who may have decisional impairment. The second pillar is beneficence, and that means that the research is designed to offer as many benefits as possible while minimizing risks, and that's an important part of study design we're going to talk about. And finally, the third pillar is justice. So the burden of research on the participant is borne, hopefully, by those who will benefit from the research. So given that framing, which the Code of Federal Regulations governs the IRB's ability to approve research, whether it's conducted under the Food and Drug Administration or the Health and Human Services Department or both. We're going to look at and use what we call the 111 criteria, which are named for the position they occupy in the Code of Federal Regulations. Fortunately, they're very, very similar and worded very similarly. So I'll use the HHS regulations, but realize that the FDA's are virtually identical. And these criteria serve us very well in thinking about safety in research and how best to protect participants. So you can see that the very criteria that the IRB uses to approve research are those that protect participants. So we're going to walk through the 111 criteria. Number one, A1, risks to subjects are minimized by using procedures that are consistent with sound research design and they do not unnecessarily expose subjects to risk, and whenever appropriate, by using procedures already being performed on the subjects for diagnostic or treatment purposes. So how can study design minimize participant risk, and how can integrating standard care and clinical care into the research protect participants? Good study design is foundational, and it ensures scientific validity, which of itself protects participants. So good study design, first of all, prevents selection bias by using strategies such as randomization, random sampling, stratified sampling, and by having clear, explicit inclusion and exclusion criteria, participants are protected. Good study design also prevents any bias that can occur with data collection, and that includes blinding, masking, placebo, and washout, different strategies. It also includes having standard research objectives and procedures that are clearly written, and finally using validated and objective outcome measures. And third, good study design prevents analytic bias. So studies must be registered prior to startup at, for example, clinicaltrials.gov. There should be transparent reporting of results. And whether you're able to prove or not prove your hypothesis, it's really important that even negative results be published. Increasing the relevance and supporting good study design means including patient participants in the research. And that can occur by using patient-centered outcome measures, by using mixed methods for outcomes, both qualitative or narrative outcomes, as well as quantitative ones. And finally, by conducting what we call participatory research, which spans around from having a community advisory board that gives input to community-based participatory research in which members of the community are actually members of the study team. How do we incorporate standard care in research? And I want to be clear that I'm using the phrasing standard care, not standard of care. Standard of care implies that there is only one evidence-based way to treat patients. And it is more helpful to recognize that for many conditions, there are several standard care plans that are appropriate and approvable. So number one, standard care in research provides a safe comparator and an excellent benchmark. So it creates a very feasible and pragmatic comparison group. Number two, it minimizes potential harm from new interventions because at least one arm of the study, those participants are getting care that we know works. So fewer participants are exposed to untested interventions. And then third, and perhaps pragmatically and most obviously, it reduces participant burden. So by taking information from procedures, whether it's blood tests or imaging that's already being conducted to care for the patient, they experience a lower burden and less risk. Okay, A2, risk to subjects are reasonable in relation to anticipated benefits, if any, to subjects and the importance of the knowledge that may be expected to result. In evaluating risks and benefits, the IRB should consider only those risks and benefits that may result from the research, as distinguished from risks and benefits of therapies that subjects would receive, even if not participating in the research. So that was a long-winded way to say that we can only consider research risks, not standard care risks. So how does the IRB evaluate risks, and how can research risk be distinguished from standard care risk? So job one, and this is often can be confusing, it can be challenging both for the study team and the IRB, is to differentiate clearly which procedures are standard care and which are research. And the protocol has to be very clear, and that has to be translated into an informed consent document that's very clear. It might sometimes it even involves putting a label, standard care research, standard care research. So it's very, very clear. Number two, we ask that the details of the risks of the research procedures are explained very clearly. So each one has to be identified and explained clearly in the informed consent. And then finally, there have to be mitigation plans for each risk. And that can be phrased, we will protect against this, whether, for example, breach of confidentiality by, for example, safe storage in a password protected secure database. So every risk, the potential participant needs to understand how that risk is mitigated. So here's an example of, it's a bit simplistic, but hopefully it gives the idea. So would endoscopy risks be considered in the protocol as a research risk? Well, the answer is it depends. So let's take a couple hypothetical protocols. Protocol number one, let's say we have patients, potential participants who are scheduled for an endoscopy, and they're invited to participate in a study that compares use of a balloon capsule with standard visualization. Okay, so in this protocol, in this instance, the potential participants must be getting an endoscopy for clinical purposes. So that's an inclusion criterion, actually. So thus, the risks of endoscopy are clinical risks and do not belong in the research-informed consent. That doesn't mean that the whoever is performing the endoscopy is not getting informed consent for the endoscopy, but those risks are in a clinical consent. Okay, now let's compare that to protocol number two. These are hypothetical ones. So let's say patients with a history of H. pylori, which is a bacterium that can cause stomach upset, gastritis. They're invited to participate in a study to look at measuring outcomes. So it's going to compare endoscopy plus blood work with blood work alone. Okay, so I think you can see in this instance, the risks of the endoscopy are research risks because one arm is getting an endoscopy and one arm is not. So the risks, all the risks of the endoscopy should be in the research-informed consent. Let's talk about risk briefly. How do we know what is risky and what's not? So the Code of Federal Regulations doesn't give us tons of guidance, but it does define minimal risk. So minimal risk means the probability and magnitude of harm and discomfort anticipated are not greater in and of themselves than those ordinarily encountered in daily life or during performance of routine physical or psychological exams. And for example, for children, minimal risk is anchored in the preventive healthcare visit, the well child care visit. Physical exam might be blood draw, surveys, etc. Who gets to decide? So anything that's not minimal risk is therefore greater than minimal risk. So who gets to decide that? So we have a range of investigators and they have a range of professional experiences. So for example, physicians, surgeons who are in the emergency room and the operating suite are the lens of their professional experience would be quite different from those of those individuals who are not in those technical, highly challenging situations with very ill patients. So the board, the IRB board, which is composed of advanced practice providers, so nurses, physicians, ethicists, community members, as well as non-scientists, is ultimately responsible for making that decision. And I think it's very helpful. The informed review includes a lay perspective. So when we talk about risk, you're probably pretty familiar with the standard risks and mitigation strategies. So I'll buzz through this quickly, but when we think about risk with a protocol, we tend to think about a biomedical protocol, and we tend to think about a new drug or biologic or device. So, for example, for a new drug or biologic, typically there would be a series of laboratory tests, as well as perhaps imaging and perhaps symptom surveys. And then there would be sort of cutoff points at which, if X, Y, or Z were noted, then either the drug dose would be reduced or the person would have to be exited, or other events would occur to protect that participant's safety. One of my favorite acronyms is DILI, drug-induced liver injury. So to protect against that, clearly the mitigation strategy is a schedule of hepatic function testing with specific cutoffs, again, upper limits for action clearly explained. If cardiac arrhythmia is potentially a risk of a device placement, there's going to be EKGs that are performed at strategic moments with content experts who've decided when it's safe to proceed and when it's not. Sensitive survey questions raise risk, and making sure that participants are aware that they can, any, and in fact, many questions can be skipped. And usually we request or require that resources be available embedded in the survey. Breach of confidentiality, we'll talk about a bit later also. But I'd also like you to consider what I call hidden risks. So these are risks that many investigators don't think about as they're submitting a protocol to the IRB. One very important risk is that formal depression screening can reveal risk of suicidality, even if that question is not directly asked. So if somebody has chest pain and they're worried they have a heart attack, pretty much everybody knows that you call 911, and there's very little stigma associated with that. You're sick, you might have a heart attack, you need to go to the emergency room. But the stigma associated with mental health illness or mental health potential harm is great in our society, and not everyone knows who to call. Not everybody knows that you can call emergency services in Frontline, for example, in Cuyahoga County. So anytime there's formal depression screening, our IRB, I'm the chair of the biomedical committees for University Hospitals Cleveland Medical Center, IRB, require a prompt and proactive mitigation strategy that usually involves outreach to the participant. Another type of risk, which actually was codified in the Code of Federal Regulations as requiring mitigation in the most recent, the 2018 changes to the common rule, are that research monitoring can accidentally, essentially, reveal incidental findings that are actionable. So, for example, imaging might discover is intended to look at the liver, but reveals something in the spleen. So there has to be in the protocol a plan, and in the informed consent, sharing of that plan in lay terms without medical jargon of what the study team will do. If we find anything that is clinically relevant on your scans, we will contact you. Another type of risk is one that most investigators don't fully appreciate, and that is when two standard care treatments are being compared in research and individuals are being randomized to one of those two. Those standard care treatment risks are actually become research risks. And the randomization procedure is another risk of the research. Ethicists argue about whether it is more risky or less risky to have your physician decide which of the two treatments or to be randomized in a research protocol. And we won't go down that rabbit hole right now. But the point is that those risks of the standard care treatments, as well as the risk of randomization, need to be clearly described in the informed consent. Genetic testing is increasingly included in many, many protocols. And while de-identified genetic information is generally submitted to databases, as medical progress gallops forward, re-identification of the individual becomes more and more possible. And it's important to notify potential participants of that risk, to make it explicit, and ideally to give them a chance to participate in the research without participating in the genetic portion if they so prefer. and if it's not core to the research aims. And then finally, I want another hidden risk is in the state of Ohio, you're an adult when you reach age 18, but as you're likely aware, 18, 19, and 20-year-olds are not legally allowed to drink alcohol or use cannabis. When surveys inquire about multiple potentially illegal activities, we have to be really careful that they advise in the survey, as well as in the informed consent, that those for whom those actions may be illegal should either consider, prefer not to respond, or skipping the question. We used to be able to obtain a certificate of confidentiality from the NIH. Right now, that process is not occurring for non-NIH funded studies. But regardless, there is legal risk potentially to participants and to anyone who endorses use of illegal substances. So take-home messages about risk. Every single research risk has to be clearly and carefully, first of all, identified and then described in lay terms without medical jargon, both in the protocol and more relevantly in the informed consent. It's important to recognize that all research does have risks, whether even a survey has risk of emotional discomfort. Anytime your data is taken and stored somewhere, there is a risk of breach of confidentiality. Even when it's de-identified, mistakes happen when identifiers could be taken. So, and finally, if risks are not known, for example, a phase 1 study in a new drug or biologic, that needs to be clearly stated. We don't know all the risks that are possible. IRB requires, and investigators are, of course, are going to provide a mitigation plan for each risk that is risk specific. So, and with a commitment to promptly inform if there's any new or unforeseen risks. And I think it's a humble reminder to all of us that even with the most comprehensive and thoughtful mitigation plans, none of us can make research completely risk-free. Okay, continuing our gallop through the 111 criteria. Selection of subjects is equitable. Third sentence, the IRB should be particularly cognizant of the special problems of research that involves a category of subjects who are vulnerable to coercion or undue influence, such as children, prisoners, individuals with impaired decision-making capacity, or economically or educationally disadvantaged persons. And the FDA specifically adds three other groups of individuals: pregnant women, handicapped, and mentally disabled persons. You may argue with the phrasing there, but that's what the FDA says. So the important point here is the phrasing "such as." So this is not a complete list of individuals who are vulnerable. And what makes a population or a group of people vulnerable? And how can we evaluate and mitigate that potential risk of coercion or undue influence? So first, it's important to realize that coercion and undue influence are actually different. So coercion is generally directed and obvious. It's the practice of persuading someone to do something by a physical or verbal threat. If you don't join this study, your benefits will be cut. So it's a very specific threat. Undue influence is generally situational and quite subtle. So it's a situation in which a person is induced to act otherwise than by their own free will or without adequate attention to potential consequences. So who are vulnerable populations? And I tried to create three groupings to have some broader overview and also to kind of free you up to realize that there may be many other populations that are vulnerable than those that are listed in the Code of Federal Regulation. So first, those who lack decisional capacity, and whether that's due to temporary, permanent, or developmental issues. That would include children, because children developmentally may not be able to protect themselves fully depending on their age and maturity. It includes or can include persons with certain mental illnesses who may be afflicted at times and unable to protect themselves fully because their thought process has been sabotaged by their mental illness. And it can include individuals with dementia or those with intellectual disability. Intellectual disability is different than lack of decisional capacity, but those individuals with intellectual disability could have lack of decisional capacity. A second big grouping is those who may be subject to undue influence due to current, historical, or societal power imbalance in relationship to researchers. So that actually includes us, okay? It includes employees, it includes trainees. We are dependent on our institution and those who grade and evaluate us, so we have a potential power imbalance there. It includes individuals who have educational or economic disadvantage who could potentially be influenced to do something, either out of lack of knowledge or desire for remuneration. It includes minoritized populations due to historical and current societal power imbalance. It includes prisoners who clearly do not have full free will. And finally, those who are not fluent in the dominant language would definitely be included here. And then the final grouping of vulnerable individuals would be those with biological or physical conditions that could interfere with self-protection or potentially make certain research procedures less safe for them. For example, pregnant persons, those who are physically handicapped, for example, a 6-minute walk test could be extremely stressful. Frailty due to either age or a chronic illness. One second, I lost my mouse for a minute there. All right, so how do we protect safety and welfare and make sure that the selection of subjects is equitable and protects them? Each of the subparts of the Code of Federal Regulations includes additional protections for pregnant women, fetuses, neonates, subpart C for prisoners, and D for minors. The entire research process actually impacts equitable selection and inclusion, and it impacts safety. So the process of recruitment, the process of enrollment, does it assess and acknowledge decisional capacity? Consent, does it incorporate the need for additional guardianship or protection? And finally, study design is very important. The inclusion and exclusion criteria need to consider the safety of potential participants, exclude those for whom the study might not be safe. And ideally, they're content experts on the study team. We actually require this through either departmental review or co-investigators. And ideally, members of the population also are on the study team who can best scrutinize what will be done. So some examples of how safety is protected through study design and through enrollment. For example, for recruitment, what we have learned from our the content expert who consults to the IRB is that particularly for gender diverse populations, those individuals are at higher risk for homelessness and for lack of access to internet. And additionally, privacy is critical. So rather than an email or a letter, which could potentially lead to a privacy breach, we encourage HIPAA protected messages that are within the electronic medical record or face-to-face recruitment. And that's what our content expert has taught us, and we're very appreciative to know that, so that we can better protect those gender diverse populations going forward. In the consent process, clearly minors are protected because parental permission is needed to approach, and there's also an assent process. And for those with possible loss of decisional capacity, for example, they're in the intensive care unit, we as an IRB have specific procedures that we outline that are approvable. It's not that we wouldn't approve other procedures, but we try to make it easy for investigators. So first of all, is that individual alert enough for a consent process? Are they comfortable enough? Are they not in pain? And are they medically stable? And if the answer to all those questions is yes, a consent process can proceed, but it has to be followed by essentially a teach back. Not a quiz, but rather a teach back, which assesses whether the study team was able to present that study in a way the individual could understand. If they can't understand, then we move to a legally authorized representative or move to returning at a different time point, and always include an assent process, which is different than the assent for minors. It's generally a couple paragraphs in very clear language that outlines procedures, risks, benefits, alternatives, and emphasizes the voluntariness. Finally, for study design for pregnant people, ideally an OBGYN is on the study team to make sure that the study procedures are aligned with the care that would be provided to a pregnant person. We've learned that for those with mental illness or substance use disorder whose capacity may fluctuate, it's really important to have the option to rescreen for eligibility to support and promote inclusion. Okay, A4 and 5. Informed consent will be sought for each prospective subject or their LAR, which is legally authorized representative, in accordance with 45 CFR 46.116, and appropriately documented or signature waived in accordance with 46.117. So how does informed consent increase safety? Informed consent operationalizes that first pillar of the Belmont report, respect for persons. And it safeguards the rights of individuals because it operationalizes the voluntariness of research. That's an essential part of the informed consent. You are being asked to participate in research. Research is voluntary. Informed consent also respects the autonomy of the individual by the decision is theirs. And you likely know that informed consent is not just a document that gets stuffed into a filing cabinet. It's actually ongoing throughout the research. So the individual knows that they have the option, if at any point the research is not right for them, they have the option to discontinue, to exit safely. A6, when appropriate, the research plan makes adequate provision for monitoring the data collected. Okay, so what does that mean? What kind of snooping are we talking about? So for greater than minimal risk studies, which are, for example, FDA regulated, perhaps phase 1, multi-site, they include masking or blinding, they include vulnerable populations, perhaps a bundle of interventions that are high risk. A data safety monitoring board or committee is generally required. It's often already been selected, as well as study team monitoring, and we request that the frequency, our lowest bar is every 4 weeks during active study conduct. For those protocols that are greater than minimal risk but lower overall risk, so maybe single site, single intervention, and the intervention is greater than minimal risk but not high risk, then a safety medical monitor, again with study team monitoring, is generally sufficient. For those studies that are not greater than minimal risk, monitoring is still needed to assure adherence to protocol and make sure that there aren't safety events occurring. This happens to be the approach that the UHCMC IRB takes, and I'm confident that other institutions take very similar approaches. So A7, when appropriate, there are adequate provisions to protect the privacy of subjects and to maintain the confidentiality of data. So how are privacy and confidentiality different? I feel like they're misunderstood siblings. So privacy is your right to be free from intrusion, interference, observation, or being disturbed, to be safe in your own space. And pragmatically, for research, that we are guided by both the Code of Federal Regulations, but also by HIPAA laws. So the IRB has to grant a pre-screening HIPAA waiver for you to get the information that you might need to screen for eligibility and reach out to potential participants. And study recruitment methods are highly scrutinized to make sure that they respect privacy. Confidentiality is after the data is collected. So it's your right to protect your personal information. And pragmatically, again, we're governed by HIPAA laws. Within the informed consent, there's a HIPAA section in which the participant gives the study team the right to access and their protected health information. They can take back that right at any time. Researchers can only request access to those elements of protected health information that are essential to answering the research question. And finally, the IRB can only approve highly secure data locations. Recruitment methods that do generally protect privacy include, first of all, in-person recruitment. If someone walks into your exam room or the room you're in and you're not interested, you can tell them to please leave. It is your space. Letters and emails that do not include protected health information, for example, hello or no names, and that do not include specific diagnosis information, and that always include the phrasing, you may or may not be eligible for this study. My chart recruitment is acceptable. It's HIPAA protected and private, and hopefully no one else is in your chart except you. Posters, flyers, and social media are things that you can look at or choose not to look at, so your privacy is protected by you. And then finally, scripted phone calls that come after initial contact with mail or email is allowed. In general, recruitment methods that do not clearly protect privacy include, first of all, cold calling. Who doesn't dislike being called in the middle of dinner for a purpose that you are not interested in? We, at least at UH, do not approve text messaging as an initial contact. Once someone is enrolled, then frequently the study team and the participant will agree to use text messaging for reminders and appointments and surveys and so on. But as an initial method of contact, it's awfully hard to completely block or silence all texts. And you don't want to silence those from your family, right? Or direct secondary recruitment. Excellent data security protects confidentiality. So each institution has its own set of data storage choices that meet security standards, that are auditable and are approvable. In general, institutional REDCap and the CWU secure research environment are approvable, but I am not speaking for other IRBs. Any data breach that releases PHI, first of all, it is not safe for participants because identity theft can literally ruin your life. And secondly, it's not safe for the covered entity because there's a huge cost associated. So now we've gone through all the A criteria of the 111 and we're on to B. And the B is when some or all of the subjects are likely to be vulnerable to coercion or undue influence, such as, and I won't read the list, additional safeguards have been included in the study to protect the rights and welfare of these subjects. And I think the Code of Federal Regulations doesn't repeat itself a lot, but it repeats itself here, driving home the point that providing safeguards is critical. And the only way we can approve a study is when there are those safeguards. And that responsibility, the shared responsibility of the investigator and the IRB, extends from pre-screening all the way through the performance of the study to continue review, write-up, publication. Okay, so let's talk about non-subjects. And you might be thinking, well, why are we talking about non-subjects? This is supposed to be about research. So the Secretary's Advisory Committee on Human Research Protections, SACRP, as we call them, actually really adds to our understanding on this topic. So how in the world can research impact non-subjects, right? So what they say is research may introduce risk to bystanders, to communities or individual subjects in which individual subjects and non-subjects reside, to research team members, and to other personnel at research sites. Harm can come from interventions or procedures, or from results or outcomes of the research. So does the IRB actually have a regulatory responsibility to protect these bystanders? And the answer appears to be yes. So in subpart A, in which the 111 criteria are located, subpart A refers to the requirement for reporting of unanticipated problems involving risks to subjects or others. So an unanticipated problem is when something is truly unexpected, when it is related to the research, and when it increases harm. That's an unanticipated problem. So the or others, clearly that's referring to non-subjects. And then subpart B requires IRB consideration of risks to fetuses or neonates in research involving those who are pregnant, in which the fetus may not actually be the subject of the research. Largely, the Code of Federal Regulations is pretty silent on non-subjects, but SACRP asserts, and I think that's a very declarative verb, that IRBs have an ethical obligation to consider the risks of research to non-subjects. So what are some examples, you may be wondering? So for example, there can be harm to sexual partners, to breastfeeding infants, and to fetuses from investigational therapies given to a subject. And so explanation and the informed consent about potential risks, and frequently collection of data from those bystanders once permission is gained can be an important mitigation. Gene therapy can cause unanticipated heritable effects. There's not a clear direct mitigation, but making others in the potential subject aware of that risk, and there may be monitoring that can be appropriate or helpful. Apparently this has occurred. There can be harm to partners of subjects living with HIV due to riskier practices from an overestimation of the efficacy of an experimental treatment. So it's very important in those situations that there's really good information in the informed consent about risk-taking and about risk, and potentially even messaging for partners. Finally, another example is caregivers of subjects who have delirium or dementia can potentially exhibit aggressive behavior if the treatment is not helpful, or if they're in a standard care arm in which their behavior is not being helped. Sometimes the caregiver actually can and does become a subject in the research, but at least there should be forewarning in the informed consent. So how can the IRB address these problems? And without a clear regulatory mandate, how are we going to avoid overreach or creep? SACCARP advises the IRB to consider three specific elements. First of all, how likely is the harm? Secondly, what is its potential magnitude? Is it great? Is it severe? And third, what's the causal proximity? So is that potential harm a direct and immediate result of the research? And they encourage us to avoid duplicative care mechanisms or services. So for example, if a local health board can be involved in the care of non-subjects, that would be appropriate. This statement is within SACCARP's guidance, and I'd like to share it with you verbatim. The U.S. government's provision of reparations to the infected partners and offspring of the subjects of the public health syphilis studies at Tuskegee and the U.S.-led syphilis experiments in Guatemala provide examples of formal recognition that the ethical responsibilities of research extend beyond those owed to research subjects alone. And I think that's a powerful statement and really drives home why we do need to consider the safety of non-subjects. So finally, let's talk briefly about us, about research team members, about protecting our own. So there are risks to being a study team member. First of all, there can be direct physical harm from a combative or very distressed participant. If the study population is at high risk for distress or aggressive behavior, then it's important that within the protocol, there's protocolized training on how to de-escalate. And even if not, it's important for study team members to have good general knowledge about how to de-escalate, to step out, step away, and call for help if there's any potential for physical risk. There's also potential harm from infectious causes, whether these are protocol related or non-protocol related. During the pandemic, COVID acquisition was certainly a risk. So if it's an infectious disease focused protocol, then good training on administration of the vaccines, the medications is very important, and use of all adherence at all times to the best hygienic practices. There are safety risks with home visiting or with other out-of-hospital contact, and we have a protocolized way to support study teams in mitigating that risk. But for example, it includes careful planning of the route ahead so somebody knows where you are, planning the parking with the participant, making sure it's a safe place, using teams of 2 whenever possible, being in close touch with the participant, and knowing that somebody expects you back by a certain time. There's also the risk of exhaustion and stress due to work burden. Maybe there really aren't enough RAs on the study. So I think that each one of us should initiate advocacy for reasonable hours, for coverage when we're sick, and for help with responsibilities when needed. And I encourage you to speak up for yourself, because we can't do our job well if we're exhausted. There can be emotional distress that's related to personal concern for a participant. So the clinical characteristics of specific participants can be very sad. It can be upsetting or triggering, whether, for example, someone who's experienced violence or a participant who has cancer. So I encourage you to support your colleagues, talk with a trusted person, and embrace professional help. And then finally, there can just be regular old interaction challenges, right? People are people. So I think that remaining polite at all times, promptly ending any interaction that feels verbally abusive, that feels denigrating, that makes you feel unsafe or degraded, you need to stop that interaction. You walk out, you leave, you call for help, you document immediately, you call for security if it's appropriate, and inform the principal investigator. This website is worth a visit. I was honestly surprised when I looked at the map that some of those red dots, they're in the US. Scholars are at risk everywhere. The safety of researchers, the principal investigators, co-investigators, for all of those in research, we can't take it for granted. What can we do? Well, we can disagree with science in scientific ways. In other words, disagree with published work in scholarly ways. Avoid personal attacks on researchers, avoid judgmental phrasing, and stand up for each other, because if any investigator is under attack, we are all under attack. Thank you very much for listening. Thank you to Dr. McComsey for all the support and guidance she provides to all of us. I'm very, very grateful for her tenure. Thanks to Malia Fink, the HRPP manager, to Beth Hackersfeld, who kindly reviewed this presentation, gave me some great ideas and pointers. And I'm probably thankfully done, and I'm happy to take any questions. I'm going to go ahead and stop sharing. Is that okay, Laura?
Sure.
Okay.
So there was a question in the chat. Can you share the website for scholars at risk?
Yes, it is right in those slides. So, but I'm happy to send it to Laura and she can disseminate it. Yep. Thank you. Oh, I see Patricia. I see a hand raised. Is that? Or not? No, maybe not.
Feel free to unmute if you have any questions or put them in the chat. What a great presentation, Dr. Furman. Thank you so much.
You're welcome.
I'm curious what, how does a patient then, if they feel like their safety has been compromised or confidentiality has been compromised as a participant, how do you report that? Are there ways where, obviously, I'm sure there's some contact number that they've been shared and they just kind of clicked and agreed and didn't necessarily read through maybe the, instructions previous to getting started. So can you talk a little bit about that?
Absolutely. It's a good question. So the Code of Federal Regulations requires that each participant receive a signed copy of the informed consent. So in that informed consent, not only is there contact information for the study team, which you can use if there's a problem and you're worried about your safety with a treatment, or you can call that number and it's there. If you're not satisfied with the study team's response, or you're worried about an overarching concern, or they haven't been able to address your concern, there is also within the informed consent, we informally refer to it as the complaint line, but it is a line that the IRB staffs for any participant who feels that their rights or their safety weren't respected or protected. And those are taken really seriously, no question. I mean, they're investigated. It is, and they're escalated to whomever could be helpful. So definitely there are, and hopefully as part of the informed consent process, the study team is making that individual aware that, hey, we're going to do everything we can to make this a great and safe experience for you. But if you have any concerns, anything is going on, call us. Here's the number. So, yeah.
Great. Thank you. Any other questions from those in attendance? I could go on and on.
You're welcome to keep asking. I mean, I recognize a lot of your names, and many of you are, highly experienced study team members. So a lot of this may be what you already know. But yeah, don't be shy about asking anything.
Yeah, we should also mention too that, at the CTSC, we do provide recruitment and study support. So anything from study design to project implementation. So If you're ready to submit to the IRB or it's in review, anything like that, we are able and prepared to help with. You know, getting access to participants and how many to get enrolled. So there are services available to CTSC members. I'll put up a QR code here that shares our link to become a member. And of course, all services at the CTSC are free and available to members. So of course, if you have questions about that, again, study design, recruitment methods, anything clinical trial related and not, we can help with that. So we do have dedicated staff that can help assist.
And I know there's also help available with the writing process, both writing the grant or the
Mhm.
proposal, as well as writing once it's done. So,
Yes, of course. And, things like vouchers, voucher funding for our members to, even use things like the mobile research unit um, for clinical trial purposes and reaching maybe hard to reach demographics of certain populations. So that's been a really great tool as well, the UH Mobile Research Unit.
for sure. The
So one of
bus.
its kind. Yeah.
Good. Right.
All right. Well, thank you so much, Dr. Furman, for taking time today and being a part of our birthday celebration. Thank you for that kind wish. And Translational Science Week continues tomorrow with the release of our theme pilot RFA. So again, sign up
Woohoo.
to become a member and hear about the next themes that will be included in that research funding opportunity. So that's up to $50,000 and I believe the range is 25 to $50,000. But as a former annual pilot awardee, Dr. Furman, we also have that application process open right now. LOIs are due in October. So please feel free to reach out with any questions and how we can support you moving forward. So thank you all so much for joining and enjoy your afternoon.
Okay, thank you so much, Laura, for running this all. All right, take care, everyone.
Bye-bye.